Girls, today I will continue with the last topic, okay? That is the role of the microorganisms in organic matter decomposition and biological nitrogen fixation. You have already studied the different types of microorganisms present in the soil. Now I will continue with their roles. Okay, their roles in the soil. What is the role of these microorganisms in the soil? So the first one which is mentioned in your syllabus is nitrogen uh, fixation, and the other one is organic matter decomposition. So I will start with this, okay girls? So before we start with this, let us try to understand the whole topic. Okay, what is nitrogen fixation? Now nitrogen, it is a critical limiting element. Okay, it is a limit, it is present in a limiting amount, a limiting element for plant growth and production. And this nitrogen, it is a major component of chlorophyll. You know what are chlorophyll, right? The most important pigment needed for photosynthesis. So the chlorophyll, which is a very important component, a very important pigment, it has nitrogen as a major component. And not only that, the amino acids, okay, which make up the proteins, the amino acids which act as the building blocks of proteins, they are also consists of nitrogen. And nitrogen is also found in other important biomolecules, okay, the molecules which are there in the biological systems in our body, in any other living organism such as ATP, and also nucleic acids like DNA, RNA. So we can see that nitrogen is a very, very important component of life, of all these biomolecules. Okay, so the even though this element, this nitrogen, it is one of the most abundant elements, we can see in the form of nitrogen gas, right? In the form of nitrogen gas, we used to write like this: two molecules of nitrogen. So this nitrogen gas is present in the Earth's atmosphere, but the plants, okay girls, the plants, they can only utilize the reduced form of this element, the reduced form. That means this nitrogen which is present in the Earth's surface, in the Earth's atmosphere, cannot be used directly by the plants. The plants can only use when this nitrogen is in its reduced form. So plant, they acquire these forms of combined nitrogen by number one, we can say by the addition of ammonia or nitrate or nitrite fertilizer, by the addition of fertilizer. Okay, when you add fertilizer in the soil which has fertilizer or manure in the soil which has these uh, compounds of nitrogen such as ammonia, nitrate or nitrite, these can be used by the plants, or the plants can also use nitrogen, can also obtain nitrogen through the release of these compounds during organic matter decomposition. We will study about this topic later, organic matter decomposition. Now, during the organic matter decomposition by the microbes, they release the nitrogen into the soil into a simpler form of nitrogen by which the plant can use. So the third one is that the conversion of the atmospheric nitrogen into compounds by natural processes. Okay, there are also natural processes by which this nitrogen can be converted into simpler form, and these natural processes are through lightning. Okay, that means the physical process. And the fourth one is biological nitrogen fixation. Okay, so here biological nitrogen fixation, bio means life, right? So you understand that biological nitrogen fixation is the fixation of the nitrogen with the help of some living organisms. Okay, so those living organisms here are nothing that we are going to talk about are the microorganisms or the microbes. Okay, so it is these microorganisms which are which help in this nitrogen fixation process. So we will not talk about these in your I will just explain on this process on the role of the microorganisms in fixing the nitrogen in nitrogen fixation. Okay, so we will talk about this one, the biological nitrogen fixation. Now, biological nitrogen fixation, I will just write in short as BNF, okay? It is discovered by scientist Bijenrik in 1901. Okay, it is found that this biological nitrogen fixation is carried out by a specialized group of prokaryotes. Okay, the prokaryotic organisms. And those prokaryotic organisms, they are the microorganisms, right? So these organisms, how they carry out this biological nitrogen fixation, before I actually start to explain to you the whole process, I will just give you a brief introduction what happened. Okay, so this biological nitrogen fixation is carried out by the microorganisms, by the prokaryotic organisms, and these organisms, they actually utilize the enzyme nitrogenase. So girls, this is a very important enzyme, okay, which has these microorganisms, they have this nitrogenase enzyme in them, which help in the process of fixing the nitrogen, the atmospheric nitrogen, of converting the atmospheric nitrogen to ammonia. Because nitrogen fixation is the process of converting a nitrogen into ammonia, atmospheric nitrogen into ammonia. So this process is carried out by the microorganisms which has this enzyme nitrogenase, and not all the microorganisms. Remember this. We will take a look at this later on, but just remember that not all the microorganisms, they has this enzyme in them. So only those microorganisms which contain this nitrogenase enzyme, they are able to fix the atmospheric nitrogen. Now the plants, they can readily assimilate the ammonia, okay, to produce the nitrogenous biomolecules, okay, such as proteins. Remember I had mentioned earlier that chlorophyll also, the nitrogen is also the main component, the major component of chlorophyll, and also it can be found in amino acids, which are the building blocks of proteins, and also ATP molecules and nucleic acids such as DNA and RNA. So all those, the plants, they can easily assimilate this ammonia, they can use this ammonia, okay, to produce these important biomolecules, okay, biomolecules. So these prokaryotes, they include the microorganisms, okay, such as cyanobacteria, or the free living soil bacteria, because we are going to talk about the microorganisms which are present in the soil. So the free living soil bacteria, and also those which can form a symbiotic relationship, okay, symbiotic relationship with the plants. So these bacteria which are free living in the soil and those which have a symbiotic relationship with the plants, they can perform this biological nitrogen fixation. So let us take a look at the whole process in detail. So here what happened in biological nitrogen fixation, okay, it can be done by the free living microorganisms, right? And the other types of microorganisms are the symbiotic microorganisms. Okay, so the free living organisms, example, I can just give you a few example like Azotobacter and the Rhodospirillum, and then we have symbiotic bacteria. The common example is the Rhizobium. Okay, and this Rhizobium, it used to form a symbiotic relationship with the leguminous plants, the legumes. Okay, so and also other types of bacteria such as cyanobacteria, such as the nostoc species, the Anabina. Okay, so here these species, bacterial species, they can fix the nitrogen. Okay, they can fix the nitrogen uh, in their free state or either by forming symbiotic relationship. Okay, so whereas these which I had already mentioned here, they used to fix nitrogen only in the free living state. That means when they are occur freely in the soil. Whereas these type of bacteria, they used to form a symbiotic relationship with the plant, leguminous plant, and they will fix the nitrogen. But these bacteria such as Nostoc and Anabina, these are the example of bacteria which can fix nitrogen in their free state, as well as in their symbiotic state. Okay, so I hope it's clear till this part. So now let us talk in detail about these Rhizobium, because these Rhizobium, they perform the very important function in fixing the nitrogenous or the nitrogen, okay, the atmospheric nitrogen. Okay, so let us talk about these Rhizobium. Now, one of the species of Rhizobium which form a symbiotic relationship with the leguminous plants, they are the Rhizobium leguminosarum. Okay, so this Rhizobium species, they used to fix the atmospheric nitrogen, okay, converting the atmospheric nitrogen into ammonia. So these Rhizobium leguminosarum, they are actually the aerobic bacteria. Okay, they are the aerobic bacteria, but they will fix the nitrogen only in the anaerobic state. Okay, they become anaerobic in nitrogen fixing state. So just remember, girls, this is very important. They are actually the aerobic bacteria. That means they perform other functions in the presence of oxygen. We call aerobic bacteria. But for fixing a nitrogen, okay, for converting the nitrogen into other forms, into ammonia, for example, ammonia, they will require the anaerobic condition, because this nitrogen fixation, it cannot happen in the presence of oxygen. So they need the anaerobic condition. So these enzymes, these microorganisms, they has an enzyme. Okay, they has an enzyme called nitrogenase, which I had already mentioned, nitrogenase, okay, in them, which this enzyme nitrogenase, it can work only in anaerobic condition. So this enzyme, it cannot work in the presence of the oxygen in aerobic condition, whereas it becomes active only in anaerobic condition. So just remember that. This enzyme nitrogenase, it has two subunits, okay? It has two subunits, the molybdenum and the iron containing protein. This nitrogenase, it is the molybdenum and iron containing protein. So I hope girls it's clear till here that these rhizobium, they has the enzyme nitrogenase, okay, which can fix nitrogen only in the anaerobic condition, okay. So only in the anaerobic condition they can fix the nitrogen, that is the conversion of nitrogen into ammonia. So here let us take a look at those process which happen during nitrogen fixation, the various steps, okay. Let us just take a look at the various steps now. So the first step here, just remember girls, is the nodule formation. Nodule formation. Now what happened? What is this nodule formation? Okay, just imagine this is the root of the leguminous plant, okay. Suppose it is the root of a leguminous plant. Now these roots, they will provide nutrition to the bacteria which are present in the soil. So by providing the nutrition, they will attract the bacteria, okay. When they attract the bacteria, this bacteria, suppose these are the root hairs, all right. So these bacteria, they will come near the root hairs. So these are the rhizobia, okay, that is the bacteria. Now these bacteria, they will attach to the root hair, okay. After attaching to the root hair, suppose this is the root hair, they will form a thread, okay. They will attach to the root hair and they will enter inside, forming the infection thread. So that is the first step, all right. They will attach to the root hair, forming the infection thread, and this thread, after the bacteria, it attaches to the root hair. Suppose this is the root hair. This root hair, it will become curl. It will become curl. Now suppose these are the bacteria, they are already attached and enter inside and forming the infection thread, okay, and they will reach up to the cortical cells. They will enter up to the cortical cells of the root. So that is the first thing here we can say, it is the formation of the infection thread. Okay, formation of the infection thread, and then these bacteria, they will enter the root through the root hairs. So the next thing is that they enters through the root hairs, okay. So next, what we have, after they already entered through the root hairs, now curling of the root hairs will take place, right? So curling of the root hair will take place, and the infection thread, it will then reaches up to the cortex, okay, up to the cortex. Then what happened, these bacteria, they will change, okay, they will change into the bacteroids. Okay, so these rhizobia which already entered up to the cortical cells here, they get modified into these type of cells called the bacteroids. Okay, so in these bacteroids, after they form inside the cell of the root, they will initiate cell division. Okay, so they will initiate cell division in the cell cortex. They will initiate cell division. Then due to cell division, the cortex will multiply, will divide and redivide. So there will be a mass of cell form, a mass of cell which form, okay, due to the cell division initiated by these bacteroids. And this mass of cell is called the nodule. Okay, it is called the nodule. So these nitrogen fixing cells, okay, the nitrogen fixing cells, they are the specialized cells that have thick wall where nitrogen fixation can take place. Okay, here these mass of cells, they are the specialized cells that have thick wall. They have thick wall where the nitrogen fixation can take place, okay, which would create an anaerobic condition for nitrogen fixation. So that means when the bacteria they enter through the root hairs, they will form the infection thread which will further infect to all the cortical cells of the root, then forming bacteroids where they will initiate cell division in the cortex of the roots. Then due to the division, repeated division of the cell cortex, they will lead to the formation of a mass of cells which form nodules. And these mass of cells, they have thick wall, okay, where the anaerobic condition is being created inside these nodules for nitrogen fixation. So girls, that is the first step, okay, the nodule formation. Now in the roots, I will show to you in the roots, in the root hairs, you will find something like this. Okay, many of these nodules formation, it's like tumor, a mass of cells which form in the roots. These are due to these bacteroids, okay, which help in the nitrogen fixation process. So that is the first step in nitrogen fixation by these Rhizobium leguminosarum. So girls, you have see here the first step, okay, in nitrogen fixation is the nodule formation, right? So what happened after these nodules has formed? When after these nodules has formed, then I had mentioned to you that they will create a mass of cell called the nodule where they will create an anaerobic condition where the nitrogen fixation will take place. So the second step here is the nitrogen fixation, okay, the second step which will occur is nitrogen fixation. Now in nitrogen fixation, I had already mentioned that it is the conversion of molecular nitrogen into ammonia, right? So now let us take a look for the conversion of this molecular nitrogen into ammonia. What are the requirements? Okay, what are the requirements which are required by these microbes, the Rhizobia microbes, to convert this molecular nitrogen into ammonia? So the first thing which is required is actually a nitrogenase, right? A nitrogenase enzyme which is present in the bacteria itself, which will help in the conversion. Now this nitrogenase enzyme, it consists of two subunits, okay, the first subunit which contain the iron, okay, protein, and the other which contain iron or molybdenum protein. So this is a non-heme molecule. So we can see that nitrogenase, it consists of two subunits, uh, and the second requirement is the pigment. Okay, this is also another very important, girls. Remember, another pigment, that is leg hemoglobin. Okay, the name of this pigment is leg hemoglobin. This leg hemoglobin is a pink color pigment, okay, which act as a oxygen scavenger. Okay, it is the oxygen scavenger, this leg hemoglobin. What it does, it remove, okay, it remove the oxygen. It remove the oxygen and it will create an anaerobic condition. Because I had mentioned to you earlier that these rhizobia, when they enter, they will form bacteroids. And in this case, actually this pigment which is present in the plants, in the leguminous plant, okay, it will create this anaerobic condition by removing the oxygen molecule. When they remove oxygen, when this condition is achieved, then only those bacteroids, they can perform this nitrogen fixation. So just remember the second very important point requirements here is the pigment leg hemoglobin. And the third one is the hydrogen donor. Okay, that means the proton donor. We need the proton donor. This proton donor can be obtained from sources like glucose, sucrose, then other sources. Okay, then we need the fourth requirements is the energy source. Energy source which can be obtained from the ATP molecules. And also we need the carbon compound which will bind to ammonia. Okay, after the formation of the ammonia, they will bind to the ammonia. So we can see here, for this nitrogen fixation to occur, we have these requirements. Okay, the nitrogenous and the nitrogenase enzymes, the pigment leg hemoglobin, which will remove the oxygen molecule, thus creating the anaerobic condition for the enzyme to be able to work. Okay, or else in the aerobic condition, in the presence of oxygen, this enzyme is not active. And also we have the proton donor, the source of energy, and the carbon compounds which will bind to ammonia. So these are the requirements. Okay, girls. So after these requirements, now let us take a look at the whole process. Okay, so if you take a look here at this whole process, this is the nitrogenase enzyme. Okay, this one here is the nitrogenase enzyme. What happened in the first process, okay, of nitrogen fixation? The atmospheric nitrogen, we know that it consists of two nitrogen molecule. We used to write like this, right? For nitrogen, it occurs in the atmosphere as two nitrogen molecule. So these nitrogen molecule, actually they have triple bonds. So just remember, the atmospheric nitrogen, it occurs in this form, two nitrogen molecule. So this nitrogen, it will attach with the nitrogenase enzyme, and in the presence of another enzyme, hydrogenase. Remember, after these nitrogen, they attach to the nitrogenase enzyme. So with the help of another enzyme, hydrogenase, these nitrogen compounds, they will get reduced. Okay, so when they get reduced, you can see here one bond here, it is removed. Okay, and now these electrons which form the bond here, they are free. So now since this nitrogen also, these two electrons here, you can see they form a bond with two hydrogen molecule. Okay, because this hydrogenase enzyme, it will provide hydrogen molecule to this nitrogen compound. So they will form NH double bond NH. So the same enzyme hydrogenase, it will again add another two hydrogen molecules. Okay, when they add to another two hydrogen molecules here, so again one bond will break from here, right? So another two electrons are free. So those two, they will form a bond with these two hydrogen molecules again. So we can see the same process it takes place, that this nitrogen, it gets reduced. Okay, so next again the last step, the same hydrogenase, it will add another two molecules of hydrogen. So finally these three bonds here are released, and this nitrogen, it forms the three bond with the three hydrogen molecules, and another nitrogen, it forms three bonds with three hydrogen molecules. So in this case we get NH3. NH3 is nothing but it is the ammonia, and we get two molecules of ammonia here. Okay, so this ammonia molecule is then accepted by the carbon compounds. Okay, and they will be used for protein synthesis. As I said in the requirements, remember we need a carbon compound which will bind to ammonia so that protein synthesis will occur. So it is in this case where they will bind. Okay, and now the enzyme nitrogenase is set free. After this whole process, it is set free. So the sum up of the complete reaction, okay, that is the anaerobic condition created by the lac hemoglobin, we can see that, sorry, it's not 2 here because already 2 here. So 2 hydrogen molecule, the nitrogen in the presence of protons provided by the hydrogenase and also the electrons. This is the same thing, okay, either this or this you can write. 8 hydrogen 8 proton molecules and 8 electron molecules with the sources of energy, that is 16 ATP molecules. They will give rise to finally two ammonia molecules. So you can see two ammonia molecule with the ATP being reduced to ADP and inorganic phosphate, and also two hydrogen molecules. So girls, it's important for you to remember this whole reaction, okay, where from two nitrogen molecules we get two ammonia molecules. So this is the conversion of the nitrogen into ammonia, that is the nitrogen fixation. Now what happened actually after this? There will be another step in the nitrogen cycle, that is nitrification. Nitrification is nothing but it is the conversion, further conversion of this ammonia into nitrites and nitrates, because the plants, they can use these nitrites and nitrates. Okay, so ammonia, it gets converted, it gets further oxidized, okay, in the presence of bacteria such as Nitrosomonas and Nitrococcus. Okay, they get converted into nitrite. Now this nitrite again, they will get oxidized by these bacteria, Nitrobacter, and they will get converted into nitrates. So these nitrates will be used up by the plants. You can see they will be used up by the plants in another process called the ammonification. Now ammonification also, in a process termed as assimilation. Okay, assimilation, where the plants, they will use this nitrate. So another step in nitrogen cycle, we have ammonification, that is the conversion of organic matter or any other nitrogenous compound present in the soil into ammonia. Okay, there is another process, ammonification, meaning the conversion of the organic matter into ammonia, and when the ammonia is obtained, then these bacteria, the same bacteria, they will convert again this ammonia into nitrates and nitrites in the process of nitrification. So this nitrogen cycle, it consists of many different steps, but you for your syllabus, okay, you don't need to remember all these things. I just mentioned here so that you will know, but we just need to study the first step in nitrogen cycle, that is nitrogen fixation. We need to know the role of the microbes. So here I'm just giving you an example of Rhizobium, okay, which are able to fix a huge amount of nitrogen, of atmospheric nitrogen, and converting it to ammonia, which is available to the plant. So you can see that they perform a very, very important role in the soil. So this is all about the first step in nitrogen cycle, that is nitrogen fixation, where we see that due to the presence of this nitrogenase enzyme, the nitrogen, it gets converted into ammonia.